commonly used for pathogen detection in humans, animals, and plants. If you look at even in the coronavirus case also, many kits are available now. Amplification, as I said, takes place at a constant temperature. So it will be economy because it does not require any sophisticated equipments. And since it uses a polymerase with the stand displacement activity, so that no tag polymerase is required. Amplification efficiency is quite high. And another beauty of this technique is can be used for even RNA templates just by addition of a reverse transcriptase. No extra time for cDNA is required. So the entire lamp takes just about 1 hour. So this is the primer I was telling about, 4 to 6 primers. So the minimum 4 primers are required for lamp. That is FIP, F3, and in the other side we have BIP and B3. And then to increase the efficiency, we can have loop primers. That is loop primer FL or BL. It can be one or it can be both the loop primers can be used. So among the 6 primers, the 4 primers are common like PCR primers. So it is the FIP and BIP that it distinguishes from the PCR. So FIP primer is quite long, about 50 to 60 nucleotide long. So as we know, it is having ambisense in orientation. That is about 50% of the bases in the sense, whereas the remaining 50% in the antisense, because this is required to form the loop at the both ends of the template. Because in the case of lamp, or template as such cannot serve as a template for the lamp. So the template has to be converted into a loop-based structure. So then only lamp reaction will initiate. Yeah, this is how a lamp reaction happens. If just for example, if you look at all the 4 primers, this is the template. All the 4 primers are added now. So let us imagine that FIP has bound here. So it's found its homologous region. So it extends the chain. So as it extends the chain, you see here F3 binds, and F3, as I said, it is a standard displacement polymerase we are using. So it dislodges this strand. So we get a single standard form like this. So this, as I said, FIP has a antisense orientation. So this will form a loop at this end, and immediately BIP will bind here, and B3 will dislodge this strand also, and you get at the end a loop-based structure or dumbbell-based structure. So this is actually the template for lamp. So if this structure is not formed, we are not going to get any lamp amplification at all. So this formation of this structure is very, very important. So that is why the 4 primers are very, very important at this stage. So once this dumbbell structure is formed, so then you will know the lamp amplification starts. So FIP and BIP can start independently the amplification. Then if you have the loop primers, they will bind at the loop region, and you will get more and more amplification. So there will be uncontrolled replication of this template. So lamp method, as we know, it produces substantial amounts of large amount of DNA. So as we all know, as the DNA is synthesized, there will be a pyrophosphate molecule released. This pyrophosphate molecule combines with the magnesium, which we supply in the reaction, forms a magnesium pyrophosphate. This, as we all know, this magnesium pyrophosphate is insoluble. So as the quantity or amount of magnesium pyrophosphate is formed, so it becomes turbid. The reaction will become turbid. So just by looking at the turbidity, we can say whether lamp is positive or negative. We need not have to even go for gel analysis or gel electrophoresis. Just by looking at the tube, we can tell. So as I said, lamp amplification requires just about 60 minutes. So look at the detection methods of the lamp. So it can be visual. So as we said here, turbidity, you can see the turbidity, no turbidity. So clearly indicate this is positive. Or to make it more subjective, we can add some coloring agents like cybergreen, ethylene bromide, or calcine. So the formation of color indicates lamp is positive. Or it can also be run on an agarose gel, and the presence of ladder-like structure indicates that lamp is positive. So this is the typical lamp reaction components. Okay, it has the thermophile buffer, magnesium sulfate, DNTP, then all the 6 primers which I said. Then betaine is required for stabilizing. Then it has a BST polymerase instead of TAC polymerase. And thermoscript RT is required if you are doing RT lamp. Okay, otherwise it is not required. So then just incubate this at about 65 degrees for 1 hour, and then we can run on the gel and see. So when you are developing lamp for the first time, one has to optimize the lamp reaction. So while optimizing, these are the parameters one has to look at. So we have to see what is the optimum concentration of magnesium sulfate required. Similarly, the concentration of butane required, units of polymerase required, and optimum temperature required. Similarly, the time required for amplification. So like PCR, a lamp also can be done using a real-time lamp machine. So then it will be about 15 to 20 minutes, we will get the results. So that's the advantage there. Coming to the second method of isothermal, that is the rolling